Now we'll continue with Lecture 4 and talk about population growth and the microbial growth curve. Microbial growth is defined as an increase in the number of cells in a population. This means that one cell becomes 2 cells, and the time that it takes for this to occur is 1 generation. The same time would be required for 2 to form 4, and so on. In each generation, cell number and cell mass double. This kind of growth pattern is referred to as exponential or log-type growth. Let's think a little bit about the math of exponential growth. When you graph exponential growth on an arithmetic scale, you see a curve such as the green one shown in this figure, shown here. Okay? When you graph this kind of growth using a logarithmic scale on the y-axis, okay, and an arithmetic scale on the x-axis, then you will see a straight line or a linear relationship as shown here in red. This is in fact the more common way to graph this type of data. It shows that there is a linear relationship between the time and cell number, or the log, more accurately time and the log of the cell number. By looking at either of this data, either in the table or the graphs, you can see that the generation time for this particular organism is 30 minutes or 0.5 hours. And I can see that pretty simply by looking up here. The time that it takes to go from 1 to 2 cells is 0.5 hours. Similar to the time that it takes to go from 4 to 8 cells, that happened between hour 1 and hour 1.5. The difference between those two is 0.5 hours. So 0.5 is G, or the generation time, also sometimes referred to as doubling time. If the scale is fine enough, you can infer generation time and some other information from a semi-log plot of this type of data. So for example, if we focus on the graph on the left, we can infer generation time by looking at the time it takes to go from 2 times 10 to the 7 cells to 4 times 10 to the 8 cells, from here to here. Since they're both 10 to the 7, we can simply compare the number in front of them to go from 2 to 4 is a doubling. How long did this take? It took from time 0, right, this part of the graph, to time 6 hours. Therefore, the generation time for that particular organism under these conditions was 6 hours. Sometimes that might be expressed in minutes, then you would just multiply by 60. The slope of the line is indicative of the shortness of the generation time. The shorter the generation time, the greater the slope. The greater the slope, the faster the organism is growing. So I can compare these two organisms really quickly and immediately see that the organism that was used to generate this graph over here in green is growing much more rapidly than the organism shown over here in red. Its generation time is going to be a lot shorter, the green versus the red. Also, please note the equation. This is an equation you're going to need to use in several problems coming up, some in your Mastering Biology homework, and some expect to be able to do this on a quiz and on your first exam. This equation here is really useful. You know what G is, it's generation time. Okay? So G is the generation time. It'll have units of minutes or hours. T is time. It's going to have units of minutes or hours. And N is the number of generations that has occurred in that particular time. It will actually be unitless. It's just a number. So if what you should do, you should really commit to memory that particular equation, and you should be comfortable with rearranging it. So if G equals T over N, then N equals T over G. Okay, and T equals G times N. Therefore, if you know any two of those parameters, you can very easily solve for the third. Okay, so there's some other useful equations that you need to know, and I've given you a couple problems here that we're going to work through. They're problems that are based on examples from a microbiology instructor at Middlebury College. Let's define a couple parameters first. N naught, okay, is the initial number of bacteria in a culture. N, and these are shown in capitals, is the number present after a period of exponential growth. All of this math that I'm going to show you is only relevant when applied to data collected during exponential microbial growth. This is a very powerful equation used by microbiologists everywhere to determine cell number and other useful parameters pertaining to microbial growth. It's the equation n equals n naught times 2 to the exponent little n. Remember that little n is the number of generations in a particular time t. Okay? So using that equation and the one from the previous slide, let's solve this problem. Let's say Bacillus cereus, a gram-positive endospore-forming bacterium, divides every 30 minutes when growing in a nice warm pot of chicken soup you forgot on your counter overnight. We're going to assume constant temperature here. If there were 100 bacterial cells in it initially, after 3 hours of exponential growth, how many bacteria will be in your soup? So there's some really important information here. 30 minutes is telling you G, okay? 3 hours of exponential growth is telling you T, okay? And 100 bacterial cells is telling you N naught. So get your pen and paper out, or pencil and paper, and let's solve this problem. So you know generation time, and it's 30 minutes. So G equals 30 minutes. Okay? So you also know the incubation time, T, in units of hours. T equals 3 hours. Generally, when we're doing math, we want similar measurements of the same property to be in the same unit. So you should begin by converting one or the other of these times into different units so that they're the same. Let's convert the generation time to hours. So G will be 0.5 hours. Okay, now these are in the same units. So now you know that G is 0.5 hours and T is 3 hours. And you can use that information in the equation G equals T over N to solve for N. Okay, which is the number of generations. So if we rearrange that equation here to solve for N, we can say that N equals T over G, which in this case is 3 hours over 0.5 hours. Okay, the units go away. That's good because I know that N is unitless and N solves as 6. So in other words, in the time that I'm interested in, which is 3 hours, there were 6 generations, or the population doubled 6 times. Okay, so now that we have that information, we can use this equation. N equals N naught times 2 to the exponent N. Okay, we just solved for this in the previous part. It's 6. All right. What about N, and what about N naught? We don't know N. That's what we're interested in, right? So we're solving for big N. We know that little n is 6. And we know that N naught, which is the initial population size, is 100 cells. Okay, so we can set this up as 100 times 2 to the 6. Okay, that equals 100 times 64. I use my calculator to calculate 2 to the 6, which is 6,400 cells. Wow, we've gone in 3 hours from 100 cells to 6,400 cells. Part of why we're doing this is to impress upon you just how rapidly bacterial growth can occur, and to think a little bit about the possible health and other consequences of that sort of microbial growth. All right, let's apply this math to another interesting and very relevant kind of problem. Let's say that we knew it took a dose of 2,000 Bacillus cereus, that should be underlined or italicized, to make you sick. This is something known as the infective dose, which is the amount of a pathogen one has to be exposed to in order to cause infection and illness, an important concept. So if that's the infective dose for this Bacillus cereus, how long would it take before you had 2,000 cells of Bacillus cereus in that pot of soup? So we have 2,000 cells, okay? That's N. We have some other information. From the previous question, we have the doubling time, G. G is 0.5 hours. Okay? So we also know the initial cell population from the previous question. We know N naught. It's 100 cells. And what we're trying to solve for is T. Okay, so that's our situation. Let's think about how we would approach it. So in this question, you want to solve for T. And in order to do that, you're going to use the equation G equals T over N, okay? Which can be rearranged to be T equals G times N, okay? So first, you have to solve for N. In order to solve for N, you're going to use the equation N equals N naught 2 to the exponent N, okay? So we're really trying to solve for that, and it's unfortunately in a rather pesky exponential form. To get it down where we can actually solve for it, we have to make it not be an exponent anymore. So this means we have to take the log of each side of the equation. We can take the log to the base 10, or we can take the natural logarithm, or ln. I'm going to stick to the log of base 10 because that fits what's in your textbook. Remember, whatever we do to one side of an equation, we have to do to the other side of an equation. So we're going to take this equation and we're going to rearrange it to solve for N, basically. So we're going to say log N equals log N naught 2 to the exponent N. Okay? And that's still kind of in a pesky and difficult to work with form. So if you remember some of the rules involved in working with logarithms, we know you'll remember that you can break this out a little bit more, the right-hand side, so that you can make that so that it is log N naught plus log 2 to the N. And we still have that pesky exponent. This is where we're going to get it down in a non-exponent form. We're going to say log N naught plus, it's a plus sign, move the exponent down, N, okay, times log 2. And now we're in business because we've got N down in a form where it's no longer an exponent and we can deal with it. Now we want to solve for N, so one of the things we can do is subtract log N naught from each side of the equation. So we can say log N minus log N naught equals N, the number of generation, times log 2. Okay, I want to get my N over on the left-hand side, so I'm going to put it over there now. N log 2 equals log N minus log N naught. Okay, and now I can divide both sides by log 2 and solve for N equals log N minus log N naught, all divided by log 2, which simplifies to log N minus log N naught divided by 0.301. And you'll see this equation written out in your textbook as well if you're not following as I'm going along. Now we're in business because we know what n is and we know what n naught is. And so if you use a calculator or Excel to solve here, you'll determine that for this particular problem is 3.301 minus 2 all divided by 0.301, okay? And that's going to equal 4.32 generations. We just solved for little n, or generation time. I'm running out of space, so I'm going to solve the rest of the problem over here. All right, but remember our goal was actually to solve for t. And now we're ready to do it, going back and using this equation. So t, or the time it takes to get those 2,000 cells, is going to be g times n. I just solved for n, okay? And I already knew g. It was 0.5. I'm going to multiply it by 4.32. And my answer is going to be 2.16 hours. Okay? Which is about 2 hours and 10 minutes. That's how much time that it takes to go from 100 cells of Bacillus cereus, of this particular Bacillus cereus, with a generation time of 0.5 hours, to go from 100 cells to 2,000 cells, which we're saying is the infective dose. So in just a couple of hours of bacterial growth, you go from having something that's safe to eat to something that's very unsafe to eat. So part of the point of doing this problem, again, is to work on your quantitative skills, to remind you hopefully of some math rules that you learned previously, and if not, you should start working on them. But it's also to give you a feeling, a hands-on feeling for the numbers involved in microbial growth and the potential implications that those numbers can hold. Now, given that, I'm not going to solve this one for you. I want you to do it on your own. Let's have a little disgusting fun. Let's say that flesh-eating Streptococcus pyogenes, a very dangerous bacterial pathogen, divides every 10 minutes at body temperature. I'm like, it's a fast-growing organism, but it's not really 10 minutes, so don't get too scared about this thing. You fall down and you scrape your knee and you get infected with 5 cells, okay, that's N naught, of Streptococcus pyogenes. After 4 hours, there's your time, okay, without medical intervention, how many bacteria will be ravaging in your body? So in other words, here, you're trying to solve for N, okay? Now once you do that, the second part of the problem is as follows. Let's say that for every 1 million bacteria, a centimeter of flesh is consumed. Okay, that's a square centimeter. After 4 hours, how much flesh have you lost? How much tissue have you lost? And are you still alive, and would you want to be? This is kind of just make it up as you go along, okay? It's the numbers that we're interested in here. So let's have you solve that on your own, please. And we can share the solution in class soon. All right, so now that we've talked about the math a little bit, let's talk about the microbial growth curve. Let's talk about the different parts of the microbial growth curve. This is one that is shown here. The red versus the green line is showing us 2 different ways to measure cell numbers. We'll talk about that in the next part of the lecture. The red line is showing us the numbers as determined using a methodology called viable count, and the green a methodology called turbidity, which is a measurement of optical density, or the penetration of light. The important thing here is to look at the shape of this graph. Please note that both scales are arithmetic. They're not logged numbers. So this is the actual data plotted in an untransformed manner. Over here, the viable count data. Over here, the optical density data. Time down here. This will be typically hours. So the important thing to start off with is that there are 4 phases of the microbial growth curve. Lag, exponential, stationary, and death. Okay? in really sophisticated textbooks, and if you go on to do graduate work, you'll talk about an additional phase called deceleration that happens right there. We're just going to stick to the basic 4, and I just wrote 3 there. Apologies. This is 4, okay? All right, what we're going to do next is we're going to talk about what is happening at each stage of cell growth. All right, so here's our microbial growth curve. Starts off in lag phase, goes into exponential phase, hits stationary, and then death phase. Okay, let's start here. So 1 is lag phase. During this part, this is a time when cells adjust to the environment. Okay, they synthesize new macro molecules, like the chromosome. Okay, so they're elongating, but not dividing, and they are metabolically active. Okay, there is no increase in cell number during lag phase, but the cells are actually doing things. They're producing new macromolecules. They're getting ready to divide. Next, let's talk about exponential phase. Okay, so it's also called log phase. And in this part we have active division. Okay, we'll usually have a uniform metabolic rate and generation time. This makes it ideal for biochemical analysis, because each cell is pretty much functionally the same at this particular time. It's a very good time to assay cells. The log phase timing is controlled by the generation time, and this is also the time in a culture which is ideal for collecting what are called primary metabolites. These are related to central metabolism. And an example of one would be ethanol production, which can be done by some bacteria and also by yeast. This is the process during, this is the time when beer is made or when wine is made. All right, so let's talk about the next 2 phases. So we just talked about lag phase, exponential phase. Let's talk about stationary phase and decline. We did this. We did this. Let's talk about stationary phase next. So during stationary phase, what is happening? From the point of view of cell numbers, cell division is equaling cell death. Therefore, there's no net change. I'm using the delta symbol there for change in cell number. Okay, that's why it's plateaued, if you look up there at the graph at number 3. Okay, so this phase is induced by loss of nutrients and accumulation of waste products, i.e. acid products. Some of them are acidic, not all of them, and others. Okay. This is a really good time if you're a scientist working with an organism. It's an ideal time for collecting what are called secondary metabolites. Examples of those are antibiotics. Secondary metabolites are things that cells make that are not the byproduct of central or energy-yielding metabolism, but that are for some other purpose, signaling, for example, as is the case for antibiotics. Lastly, we have death phase. Okay, and the math of death phase is exponential. It's an exponential decline in cell number. Okay, and what's happening here is the rate of cell death is greater than the rate of cell division. Okay? And this happens when when nutrients are exhausted. By that I mean they've run out, not that they're very, very tired. And also waste has become toxic. So if you think about all 4 phases of the microbial growth curve, okay, the only exponential ones are 2, which is called exponential phase, and 4. The math that we discussed in the earlier section of this lecture portion is all relevant to 2, although technically it can be done on stage 4 as well. That's just simply rarely or never done. It's usually always done data collected during exponential cell growth, because that's what people are generally most interested in, is how quickly organisms can grow. So those are the 4 phases of a microbial growth curve. So one other thing that I want to touch on this portion of the lecture is the idea of growth in chemostats. So to back up a little bit, the microbial growth curve that we just looked at, that kind of growth pattern only happens in basically fixed conditions. So for example, that's exactly the kind of situation you would see if you were to grow some bacteria in a culture flask where it's confined. You start with a certain amount of nutrients, they use them up, it runs out, and that drives them through the various stages of population growth that are depicted in the microbial growth curve we just examined. A similar scenario can happen to an organism growing in one part of the body, which will then ultimately function like a flask or a test tube. Now in industry, we often want to keep microbes in a particular stage of growth, most typically exponential. Okay, so we grow them in things that are called chemostats. So these are often used to keep microbes in exponential growth. And you might want to do that if you were in a pharmaceutical company or in a company that makes fine chemicals, for example, so that you could collect a maximal amount of some particular primary metabolite. You're using them to make something. So a chemostat is a large culture reservoir, and the purpose of the chemostat is to maintain constant conditions. So you're always adding new nutrients and removing waste. You're also going to maintain the pH carefully, the oxygen status, and the temperature. The goal is to maintain constant conditions, keeping microbes in a permanent sort of exponential growth phase. Chemo Stat's use is something that is taught in graduate programs in areas of fermentation biology. The knowledge to operate a Chemo Stat is highly sought after, and in fact very few people are good at it. It requires a very solid understanding of mathematics and engineering principles, and in fact many of the microbiologists that do this kind of work have engineering degrees or considerable engineering training so that they have the mathematical skills and the technical skills required to do this. Some of them, however, just have more basic microbiology training, and then they rely on teammates who have engineering skills to help them in their experimental design and in process management. And that concludes this part of the lecture.